Bismillahirrahmanirrahim. Dear students, have a nice day. Hope you will be fine, best of your health. This is Dr. Sajid Mahmud Rao, Division of Science and Technology, University of Education, Lahore. Today we are talking about steroidogenesis and some structures of common steroids, in which we will discuss the short question answers, which is relevant to the course of natural product, that is Chem 4119. Let's see the short question number 2. What do you know about the sources, solubility, and therapeutic effect of cortisol, estradiol, and progesterones? How testosterone and estradiol are synthesized? You can see over here some important common structures of hormones, and these hormones are cortisol, progesterone, estradiol, aldosterone, testosterone, and dihydrotestosterone. Let's see the sources, its uses, and solubility of these molecules respectively. The first molecule is cortisol. It is produced in many animals, mainly by the zona fasciculata of the adrenal cortex in the adrenal glands. It is soluble in water, ethanol, and isopropanol. Cortisol regulates a wide range of vital processes throughout the body, including metabolism and the immune response. It also has a very important role in the helping the body respond to the stress. Cortisol has a molecular formula that is C21H30O5, means there is the presence of total number of carbon atoms 21 and hydrogen 30 and total number of oxygen atoms that are 5. The molar mass of cortisol is 362.46 gram per mole. The next molecule is progesterone. When we see the structure of this molecule, we observe the presence of carbonyl group at carbon 3 position and acetoxy group at carbon 17 positions, as well as a double bond is present at carbon 4 position. The chemical formula of this molecule is carbon 21, hydrogen 30 and oxygen 2, means there is a presence of total number of carbon atoms 21 and hydrogen 30 and total number of oxygen atoms are 2, that is presence at carbon 3 and carbon 17 positions with acetoxy group. The molar mass of progesterone is 314.469 gram per mole and melting point is 126. Progesterone can be used as an administrative route, that is taken by mouth. It can also be taken as topical transdermal as well as vaginal and intramuscular injection. It can also be used as a subcutaneous injection and a subcutaneous implant. Let's see the sources of this molecule. Progesterone can be isolated from beans, broccoli, Brussels, sprouts, cabbage, cauliflower, kale, nuts, and pumpkin. This hormone is produced in the ovaries and the placenta when a woman gets pregnant and the adrenal glands. Progesterone is soluble in organic solvents such as acetonitrile, ethanol, and methanol. Progesterone prepares the endometrium for the potentials of pregnancy after ovulation. It triggers the lining to the thicken to accept a fertilized egg. It also prohibits the muscles contractions in the uterus that would cause the body to reject an egg. The next molecule is estradiol. When we see the structure of this molecule, it consists of hydroxyl group at carbon 3 position and the other hydroxyl group at carbon 17 position. The total number of rings in these molecules are 4. 3 rings are hexagonal and the 5th ring is pentagonal. However, the first ring is an aromatic. The chemical formula of estradiol is C18H24O2, means it comprises total number of carbon atoms are 18 and hydrogens are 24 and oxygen atoms are 2. The molar mass of this molecule is 272.38 gram per mole, and this molecule can be used as an administrative dosage. It can be taken as an oral, sublingual, as well as intratropical, transdermal, vaginal, intramuscular, subcutaneous, and subdermal implants. Estradiol is also known as E2, and also spelled with estradiol, is an estrogen steroid hormone and the major female sex hormones. It is involved in the regulations of the estrus and menstrual female reproductive cycles. Estradiol is responsible for the development of female secondary sexual characteristics such as the breasts, widening of the hips, and the female associated pattern of fat distributions. Now we discuss the sources, uses, and solubility of estradiol. It is produced especially within the follicles of the ovaries, but also in other tissues including the testicles, the adrenal glands, fat, liver, the breasts, and the brain. Estradiol is produced in the body from cholesterol through a series of reactions and as an intermediate, as you have seen in the steroidogenesis. It can be soluble in dimethyl sulfur oxide, that is DMSO, and dimethylfuramide, that is abbreviated as DMF, and in ethanol. When we see its uses, estradiol regulates many processes in the body. Estradiol is used to treat menopause symptoms such as hot flashes and vaginal changes, and to prevent osteoporosis, bone loss in the menopausal conditions of the women. Let's see the synthesis of androgens. Androgens are synthesized and secreted primarily by the Leydig cells of the testes, thecal cells of the ovary, and cells in the reticularis regions of the adrenals. In most tetrapod vertebrates, testosterone is the dominant circulating androgen. Testicular synthesis and secretions of the testosterone is stimulated by circulating LH, which upregulates the amount of 17-alpha hydroxylase, that is carbon 17 carbon 20 lyase enzyme, a rate limiting enzyme for convergence of carbon 21 into carbon 19 steroids. Once taken up by target tissues, testosterone can be reduced by 5-alpha reductase to shield a more active androgen metabolite, 5-alpha dihydrotestosterone, that is generally represented as 5-ADHT. Testosterone and androstenedione can also be converted into estrogens, such as 17-beta estradiol, that is E2, or estrone, through a process termed aromatization. Aromatization is carried out by a cytochrome, that is P450 aromatase enzyme, that is expressed in the granulosa cells of the ovary. Leydig cells of the testes and many other tissues, including the placenta, brain, pituitary glands, liver, and adipose tissues. Indeed, many of the effects of circulating testosterone are owing to convergence into either 5-alpha dihydrotestosterone or E2 within the target tissues. Let's see the synthesis of testosterone, because testosterone falls in androgens because this is the big examples of androgen steroids. So let's see the first step in which 17-hydroxypregnenolone is converted into dehydroepiandrosterone in the presence of 17-alpha hydroxylase 1720 lyase enzymes. So you can see over here in the first reaction, you can see the structure of 17-hydroxypregnenolone in which the hydroxyl group is present at carbon 3 position and acetyl group is present at carbon 17 position. In addition, one hydroxyl group is present as alpha in the 17 position too. So this structure is converted into dehydroepiandrosterone in the presence of enzyme 17 alpha hydroxylase 1720 and lyase. The structures of dehydroepiandrosterone consist of two functional moieties. One is hydroxyl group at carbon 3 position and the other is carbonyl group at carbon 17 positions by the additions of double bond at carbon 4 position. So when we see the difference between the structures of these two molecules, you can see over here there is a presence of just 17 positions carbonyl and hydroxyl group at carbon 3 positions and double bond at carbon 4 positions in the first intermediate molecules. In the second step, dehydroepiandrosterone is converted into androstenedione in the presence of 3-beta hydroxysteroids dehydrogenase enzyme. So the androstenedione consists of the carbonyl group at carbon 3 position and double bond at carbon 4 positions. In addition, carbonyl group at carbon 17 position. So just only the changes that takes place at carbon 3 positions is the presence of carbonyl group at carbon 3 positions in the androstenedione instead of hydroxyl group, which was present in the previous structures in dehydroepiandrosterone. In third step, androstenedione is converted into testosterone, which is our target compound for the synthesis of androgens or testosterone in the presence of 17-beta hydroxysteroid dehydrogenase enzyme, which is abbreviated as 17-BHSD. So you can see in this reaction the androstenedione is converted in testosterone. In testosterone, there is a presence of carbonyl group at carbon 3 positions and double bond at carbon 4 position similar as that of androstenedione. But the carbonyl group which was present at carbon 17 position is converted into hydroxyl group as beta. So the difference between testosterone and androstenedione is just there is the presence of hydroxyl group at carbon 17 position in testosterone as compared to carbonyl group at 17 positions in androstenedione. So this is the general method for the synthesis of testosterone. Synthesis of estrogens. Estrogens and progestins are synthesized and secreted primarily by maturing follicles, corpora lutea of ovaries, and the placenta during pregnancy. The predominant secreted is E2 and the predominant progestins is progesterones. The profile of the synthesis of estrogen changes during the course of folliculogenesis, during which, under the influence of LH, the thecal cells synthesized and secrete androstenedione and testosterone, which diffuse across the basement membrane and are subsequently aromatized to estrone and E2 respectively by the granulosa cells. The level of aromatase and hence estrogens produced in the granulosa cells is under the control of FSH during mid-follicular phases. Later in the cycle, the follicle corpora lutea express greater numbers of LH receptors and LH begins to regulate E2 production. During pregnancy, the placenta utilizes androgens precursors from the fetal adrenal glands and secretes large amount of E2. In addition, in male vertebrates, many target tissues such as pituitary cells and hypothalamic neurons convert circulating testosterone into E2 steroids. Let's see the synthesis of estradiol. Estradiol is the common example of estrogens. In the first step, 11-hydroxypregnenolone is converted into dehydroepiandrosterone in the presence of 17-alpha hydroxylase 1720 lyase enzyme, which is then converted into androstenedione in the presence of 3-beta hydroxysteroid dehydrogenase. So you can see over here in this reaction, the first structure is the 7-hydroxypregnenolone, which have hydroxyl group at carbon 3 position, acetyl group at carbon 17 positions, and hydroxyl group as alpha also present at carbon 17 position. In addition, there is a presence of double bond at carbon 5 position. This molecule is converted into dehydroepiandrosterone, in which the acetyl group and the hydroxyl group, which is present at carbon 17 position in the previous molecule, is converted into carbonyl group. So this molecule is known as dehydroepiandrosterone, which is further converted into the next molecule by the effect of enzyme 3-BHSD. In second step, androstenedione is converted into estrone in the presence of aromatase enzyme, which is abbreviated as CYP19. Androstenedione is converted into estrone. You can see in this reaction, the difference between these two structures is the presence of first aromatic ring in the estrone, which is not present in the previous molecule. So there is also a change at carbon 3 positions because the carbonyl group is present in the androstenedione. However, there is a presence of hydroxyl group at carbon 3 position in the estrone. In the third step, the estrone is rearranged into estradiol in the presence of 3-beta hydroxysteroid dehydrogenase, which is abbreviated as 17-BHSD. The estrone, which has aromatic ring in the steroidal structure, which is the first ring which have the hydroxyl group at carbon 3 positions and carbonyl group at carbon 17 position, is converted into estradiol. In estradiol, there is a presence of two hydroxyl group. As the name implies, diol, one hydroxyl group is present at carbon 3 positions and the other is present at carbon 17 position. In this way, the estradiol is synthesized by using this reaction scheme.